Well, thank you so much for that warm introduction, and it's a privilege to virtually be here. I just want to make sure y'all can hear me okay before I keep going.
Yep.
Great.
Wonderful.
As you heard, my name is Ellie Van Allen. I'm a medical oncologist at Dana-Farber, where I see mostly prostate cancer patients clinically, but I spend most of my time doing research, where I run my own research lab at the intersection of cancer genomics, computational biology, and AI. And I run a research division called Population Sciences, which has about 40 or so investigators, each pursuing different kinds of, I'd say, dry lab research, which could be computer science, computational biology, could be outcomes research, and other kinds of things. For today, I thought I would present on some of our work involving AI and precision medicine. But here are also my lab website and social media handles for the future. Here are my disclosures. So to set the stage, what I thought I would do is start by defining what the precision cancer medicine challenge is to me, in one very long sentence that I will break up into pieces. So the idea starts out as a simple one. How do you translate all prior knowledge, so everything that we know about cancer genomics, biology, medicine, everything, and all knowledge about one cancer patient at one point in time, all the molecular information we can generate from a patient, other clinical data, how we can contextualize that, and use it to make the right choice for the right patient at the right time. This is at the core of what precision oncology is and has been something we've been thinking about now for over a decade on how to do better and better going forward. Um, and so there's lots of ways to think about it, but I thought I would take that very long sentence and break it up into three parts and sort of talk about some of the work we're doing in each of these three parts and where AI is increasingly potentially of interest and of use in various domains. So the first part of the challenge of the sentence of the Precision Cancer Medicine Challenge is how do we translate all prior knowledge? How do we come up with some sort of useful representation of all of the data in the universe, or maybe a little bit more refined, all of the molecular data in the universe, so that we can ground everything that we do in some sort of prior information. And we've been thinking about this for a while, and we've tried a bunch of non-AI approaches. And so one very concrete example started out with this study. So this was a study we published now a few years ago, whereby we were able to aggregate over 1,000 prostate cancer exomes. We did harmonized molecular analysis followed by mutational significance analysis and generated this, what we called a long tail of significantly mutated genes in prostate cancer. And if you scan the x-axis, you might see some genes that look familiar to you. Of note, this was a cohort that was split between primary and metastatic or lethal prostate cancer. And through this study, we did a bunch of explorations of some of these genes, what they might mean, and so on and so forth. But that's not actually why I wanted to show this. What I wanted to show was the last figure in the study. In that study, and because I think it really exemplifies this challenge of how do you represent all this information in a way that's meaningful and useful? If, for instance, what you care about is what's the difference between primary and lethal prostate cancer? And so in this last figure, we try to organize that data to ask that question. And so the figure is sort of visualized as follows. There's frequency of mutations on the x-axis in the metastatic samples, frequency of mutations on primary prostate cancer on the y-axis, and each gene here is a dot. And so not surprisingly, we were able to recover that AR, P53, and P10 were enriched in a lethal prostate cancer. This was not something we discovered. This is something that's been known for decades. But where we really struggled was how to make sense of what, if any, of the signal in here mattered. How did this form of prior knowledge, which of any of it was useful for precision oncology? And we asked whether there was a smarter or better way to dissect this signal and use some emerging principles in AI to help us understand this better. And so what we ended up doing was building something called PNET, which is a sparse neural network whereby we engineered biology into the architecture of the neural net. And so the way it works is as follows. It could take in as features any number of molecular data. So it was not limited to one data modality. So in this case, for the toy model that we published on, we had mutations and copy number alterations, but you could add fusions or methylation data, expression, whatever. And instead of having this be a traditional convolutional neural net, we sort of reverse engineered biology into it such that each node and edge had a meaning to it, and that it was representative of biology, which I'll come back to a little bit later, so that we could maximize the sort of predictive potential of such an approach from a modeling standpoint, basically get as good of a model as we could get, while also enable interpretability, so that we could open this sort of box, the model, inspect each of the layers, generate hypotheses that we could test experimentally and clinically, and then use this to better answer our questions. And so we steered this to prostate cancer, and it turned out it worked. I was sort of skipping over some of the clinical, the sort of the model performance results, but I can come back to them a little bit later if people are interested. But it worked, needless to say. And so that gave us confidence that we wanted to inspect the model and understand why it was making these kinds of predictions. And so we looked first at here are the different features that were fed into the model. And this was the first layer in the neural net, the gene layer. And as you can see, there's AR and P53 and P10. So that's good. That means it's recovering the signal we know should be there. And then what you can do is start to inspect and explore each part of the model and how it gets propagated over the course of the network towards the outcome, which in this case is trying to distinguish primary and lethal prostate cancer and the molecular signatures and features that matter in that context across different feature modalities. And so then we, instead of using artificial intelligence, used human intelligence and stared at this for a while until we noticed that, well, okay, there's P53. That's not surprising, but it's also not very interesting. We were actually quite surprised to see MDM4 pop up as a signal here. And MDM4 is a known regulator of P53. MDM4 focal amplifications, as it turned out, were highly enriched in the advanced prostate cancer setting. And we can see this signal manifest itself all the way down the model, which gave us confidence that there might actually be something real here, given the convergent biology. And as it happens, and as was shown in this particular study, we subsequently did experimental validation, including both gene editing approaches and also a use of an MDM4 selective inhibitor, and have since actually explored the use of MDM4 selective inhibitors in prostate cancer patients via a phase 1 clinical trial that we're hoping to pursue in more detail. Unfortunately, the company that we were working with to do this went bankrupt during the development process, and so we're trying to find another one. In case anyone is interested in this, please let me know as an aside. So that was sort of a very brief summary of sort of one way in which we were trying to represent all prior knowledge using AI and steer it towards a specific kind of precision medicine question. However, there were some fundamental limitations to that kind of an approach that are illustrated by this graph. So the way we encoded prior knowledge was with the use of what's called the Reactome database. It's a very convenient structured database that has relationships between different genes and how they converge into signaling pathways and processes and so on and so forth, and that are visualized in this little schematic here. But this immediately demonstrates at least two problems fundamentally with this strategy when it comes to trying to maximize, well, the learning one can do with these kinds of approaches. One is that Reactome only has about 4,000 to 5,000 genes. So we're already immediately ignoring effectively 3/4 of the genome when we apply something like this to this domain. And two, Reactome is imperfect. And I'm sure if you focus on any one of these biological processes that are depicted here and you know the biology in and out, you could go through Reactome, find something that you disagree with, and then wonder how good is this as a proper representation of all prior knowledge in and of itself. And so what we're now trying to do is go back to the drawing board and to take this schematic and basically replace the entire concept with a variety of other approaches that are still works in progress. So one thing we're trying to do is experiment with different forms of biological priors. And specifically, what we're interested in doing is pursuing agentic AI and agentic reasoning using large language models and the ability to access vastly larger quantities of information to inform prior biology. We're experimenting with newer and more advanced architectures, so graph neural nets in particular, something we've been playing around with for this context. And we're also exploring the use of foundation models as pre-training modalities to help guide sort of a better baseline for what molecular signals we're actually looking for, in addition to adding in all sorts of other kinds of modalities that we tend to see in clinical settings, like pathology, radiology, and so on and so forth, to then steer towards these outcomes. So just as a quick aside, because this stuff still works in progress, we're pretty excited about this notion of scientific agentic AI for enhanced priors. Effectively is swapping out Reactome and swapping in these tools. If you don't know what these are, the idea here is that these are conversable systems that are capable of reflective learning and reasoning. They coordinate large language models, machine learning tools, experimental platforms, or combinations therein. And the goal is that actually these things can be inserted for hypothesis generation, reasoning, learning, and discovery, in that they actually represent, effectively every scientific study and the associated logic underneath that's ever been done in history prior to the point of us doing this experiment. There's a very nice review about this topic from Marinka Zitnik and colleagues that I have cited down below. And the part of it, and this sort of graphic illustrates all the different things that agents can do. But for us, the thing we really are excited about, in spite of the rapid hype cycle, is this reasoning thing. And so I don't actually have results yet for you. This is still a work in progress in terms of how we're incorporating it into these models. But perhaps this time next year, maybe we'll have something more robust to share, and would love to do so, and disseminate what we're trying to do in these contexts. And so we want to make better biological priors. And so I think language models and their sort of agentic power could help us from a reasoning perspective. The other thing we want to do is actually incorporate vast quantities of other kinds of prior information that are not necessarily represented in that way, but are rather part of these massive single cell foundation models. And so we have been actually experimenting with a variety of the sort of existing single cell foundation models, which are largely not inclusive of cancer data, but some of them have a little bit of cancer data, but they're dominated by not cancer data, and have asked systematically whether these actually can be helpful for asking cancer-specific questions, like take single cell cancer data from patients who are responders and non-responders, and does the use of this is the use of these foundation models help to stratify or this and that, whatever. And the long and short of it is, and this is work that we presented at AACR, when we're trying to wrap up a preprint, which we'll hopefully have posted soon, is that as is the case when people have been evaluating these single cell foundation models for other purposes outside of cancer, so it is true that within cancer, it's still not clear if and how useful these things actually are. They might be helpful for cell annotation. I will say that they have been less useful as fine-tuning tasks for harder things like predicting response or clinical stage or treatment effect. I suspect we need more data and possibly newer methods, but even here, I'm not entirely sure. Some of you may have seen, and I realize this might be not the best thing to show in a session that's supposed to be about AI, but there's this paper got a lot of recent attention because it was demonstrating how a lot of these in a slightly different context, deep learning-based approaches didn't outperform simple models, at least currently. Now, the counter to that, of course, is that that's what people said about language models, 5, 10 years ago, and it just was a matter of scale and time and some creativity on methods, and so perhaps that is the case here. So that's sort of where we're at the moment in terms of translating prior knowledge and representing this. What about all knowledge about one cancer patient at one point in time? What are the challenges and opportunities there, and where are there opportunities to use AI in various ways to help inform this and squeeze as much information out of a patient's, let's say, genome or clinical data as we possibly can? This turns out is a really hard problem, and increasingly so. And there's many barriers to doing proper interpretation for a single patient's molecular data that I'll illustrate here. One of the challenges is that the technology in the clinic is evolving at a far more rapid pace than our algorithms can keep up. So when I started doing this, 10 years ago, we were doing DNA-only sequencing of small sets of genes. Now clinically, we're doing exomes and transcriptomes on all of our patients. And we could imagine a world where we'll be doing even more high-dimensional data generation for each patient at the point of care. And so we need algorithms that can mine not just simple first order relationships, like EGFR mutation, EGFR inhibitor, but more complicated kinds of patterns, germline BRCA mutation, somatic homologous recombination deficiency in tandem equals PARP inhibitor for prostate cancer. Those kinds of things are going to be harder to figure out. The knowledge about what matters clinically keeps evolving. As just in a concrete example of that, about 8-9 years ago, we actually published a knowledge base for precision medicine, precision cancer medicine, and we recently revisited it and checked and see how much of that information is still useful in 2025. And it turned out about half of the information was wrong, and we were missing a ton of new information that had actually come. And so just keeping up with all of this has been very hard. The last thing, which I won't talk about in more detail other than a third of the slide, is that we have to remember when it comes to precision medicine, there are elements of user interface and user interaction that we take for granted in other parts of our world. We don't think twice about the sort of ease of use of ChatGPT or Gemini or whatever. And in the flip side, we're giving clinicians these extremely confusing and jargon-filled molecular reports to use for an individual patient. And there's ways, and I've sort of listed some of the example citations below, but there's a lot of ways we're trying to address that kind of issue that I don't, that's out of scope for this talk, but just want to acknowledge that that's an issue. So on the first part, what are ways we can do a better job of mining more information out of our patients' individual high-dimensional data? One example is to do a better job of finding some of these complex relationships, these second-order associations like I observed. And there's a lot of them. But even within one of them, actually, this is where deep learning could be useful. And so we, not too long ago, were looking at on the germline whether there was better ways to find pathogenic germline mutations in cancer patient data. And we compared existing approaches and deep variant, which was the deep learning model put out by Google to search for germline pathogenic mutations, and found surprisingly quite a few examples in a large corpus of data where the deep learning approach was able to recover definitely bona fide germline pathogenic mutations in known cancer genes that was missed by the existing approaches that are used in a lot of clinical and research labs, and sort of illustrated early on for us where some of these AI tools may be useful. What we'd like to do, though, is sort of take a step further and incorporate other kinds of ways of maximizing the understanding of an individual patient's genomics for point of care decision making. And so something we tried to do a few years ago towards that end was to add more functional knowledge about sort of the functional readouts of molecular data. And what I mean by that is we borrowed a concept from Anand Goldenberg, who's a machine learning scientist in Toronto, who built an algorithm called similarity network fusion, which is sort of an early precursor to some of the more recent neural net approaches. And the idea here was we basically can come up with a network to match an individual patient's molecular profile to any number of preclinical models to basically try to find which preclinical models were most similar molecularly to my patient. And if you can find that, then you can look and see what are the treatments that were effective in those preclinical models and use that information for your patient. So that was the idea. As indicated in the study that's cited on the bottom left, if I'm being perfectly honest with you all, it didn't work very well. Nothing did. And I think the bigger fundamental issue is that we were trying to match patients to model systems when what we really needed to do was actually match patients to patients. And if only we had a massive set of experiences of patients that came before our patient, and we could figure out how similar our one patient was molecularly to a neighborhood of other patients like them or not like them, figure out what drugs work for those patients to tell the provider, that might be better. And so what we've since done, and this is work that's in prep, and so we're not quite done yet, but we're working on it, is created a patient similarity deep learning approach to link genomics with clinical data to be enabled to infuse real-time knowledge for individual patient decision making. And so the idea is that we took tens of thousands of patients from Dana-Farber and created genomic embeddings for all these patients. We clustered these into groups, which ended up, interestingly, recovering a variety of known kinds of clusters. So, for instance, you could find like the EGFR non-small cell lung cancer cluster, or the RBP10 deleted prostate cancer cluster. But you also could actually find some other ones that were surprising or were refined subclusters within them that have more subtle molecular findings that perhaps could yield different clinical trends. And so for all of those thousands and thousands of patients, we also used language models to infer and pull out their complete clinical trajectories, because we had access to the entire electronic health record for these patients, including which drugs they got and what happened to them. And what we've constructed now is this notion of patient neighborhoods, so that when a new patient comes in, we can put them into this latent space, find patients that are similar to this patient, and then report out what happened to these neighbors, what medications did they get, what diagnoses did they get. And so we have some vignettes about cancers of unknown primary and what are the most similar or not dissimilar patients and what drugs do they get, which are useful for both diagnostic and predictive purposes. This too is a work in progress and sort of not quite ready for prime time, but we're hopeful we can wrap this up soon, and would love to share this going forward. But I at least conceptually wanted to put it out there because I think this is not something we've seen before. But the idea, of course, is that if this can be embedded into a hospital system, it could just constantly be running and updating in real time, which would be kind of a cool way of doing real-time learning. So that's a work in progress. And the last part is we also want to make sure we're not just thinking about genomic data when we think about individual patients. And so, like many other people, we've been also interested in applying computer vision AI to cancer use cases. So this was just one example from our group, whereby we took, and this was kidney cancer focused, where we had H&Es. We could link that with associated mutation information to create a graph representation of the tumor grades and create and infer some topologies in that space, define some subsets of tumors that exhibited this pattern that we called microheterogeneity, where they had different kinds of nuclear grades interacting with each other in two-dimensional space, also infer features of the immune microenvironment, and combine these two pieces of information, in this case validating in Checkmate 025, which is a randomized clinical trial of kidney cancer patients who got either nivolumab or everolimus, and find a deep learning-based readout to help stratify which patients are most likely to respond to immunotherapy directly solely from the H&E two-dimensional image. And so on its own, it's already powerful, but we think in tandem with molecular and clinical data, we can really do multimodal learning going forward. So all of this is also to be continued, but it's stuff we're pretty excited about. And so if we first want to translate all prior knowledge, and we also want to maximize everything we can make about all knowledge from one patient at one point in time, ultimately we need to figure out a way to package all of this together so that we can make an optimal clinical decision. And so for us, what that has meant is we've been building algorithms that can be deployable anywhere, that do this for single patients or groups of patients. And so this is something called the Molecular Oncology Almanac, or moalmanac.org, built by Brendan Reardon, pictured below. And so right now, the way it works is you can take in germline and somatic molecular data and RNA and DNA information. It will do various kinds of annotation, first and second order inference, patient to cell line matching currently, although we're migrating to patient matchmaking, and as I alluded to, generate a report that we think uses some of the first principles of user interface and design that I think were lacking from the first wave of precision medicine studies. This resource is what we call sort of aggressively open source. So we have a browser, we have an API, the code is fully available. We have a portal whereby you can if you have Google Cloud credits, you can upload your data and run the stuff without us ever touching your information. And we've tried to make this as accessible as possible. And more recently, we've also sort of rethought how we're presenting this information. And so this is a study for which we already have a preprint out, but we're still working on the details. But we added, we basically took that knowledge base and used it effectively as a RAG for a language model and converted a static website into a conversible system that we can then have doctors start to query in real time, more like they're going to be using these technologies for everything else, augmented with knowledge, very bespoke and curated knowledge about precision oncology, so they can ask questions like the one pictured here in free text form and get the right answer, and not get wrong or bad information as well. We're also thinking about ways of enhancing this further so we can have region-specific instances of this whole platform. And so there are currently extraordinarily, suboptimal biases about the way a lot of these tools are built, including ours, which tend to be biased towards, for instance, only FDA indications. And so we've established collaborations thus far with a variety of organizations to try to make region-specific instances that can be queryable and are tailored to where you live. And so we have one for Europe generally, Ireland specifically, one for Canada. We're working on one for Chile. And with this, actually, we also will use AI in a very different way than we ever would have imagined. In this case, as a translator for sort of like real-time optimized translation services, so that anything we produce can be read and consumed by people wherever they live. And of course, we're always eager to engage and work with other folks near and far. And so this is a very shameless plug in case anyone is interested in collaborating with your works. We'd be very happy to have those conversations as well. And so finally, we also care about making sure this works in real life. And so we've thus far, probably our biggest collaboration is with the Veterans Affairs National Precision Oncology Program. So in the United States, the veterans, the military veterans have their own independent hospital system that's nationwide, and they have their own precision oncology program that goes with it. And our molecular oncology almanac has been built into this. And we've been testing and exploring how we can use this in collaboration with the Veterans Affairs clinicians to enhance and find more actionable mutations for our veterans, which we think is pretty cool. We haven't quite figured out how to do a prospective trial. I'm still not sure how that's going to work, especially given how rapidly these things are evolving, but we're certainly happy to explore going forward. And then finally, all of this only works if we have an engine of translational and preclinical molecular data that we can be constantly generating from and learning from our patients. And so via some of the other hats that I wear institutionally, the Population Sciences, Center for Cancer Genomics, we historically have been getting pre-on and post biopsies and doing bulk sequencing, but like everyone else, we're now doing single cell and spatial, trying to generate models and apply it to as many clinical contexts as we can get our hands on and we can get funding for, which is an increasingly complicated thing for us as well. So with that, I will end. Thank you again for this invitation. I'm sorry I could not be there in person, but obviously want to thank all the great folks in the lab and in our environment that we get to work with to do all this stuff. And I'm happy to take any questions.
Okay, thank you. Thank you for your wonderful and insightful talks. And there is one question already in online. I'll read it for you. Professor Ben Allen, thank you for the insightful lecture. It was interesting to hear your work in progress on genomic embeddings of cancer patients into the latent space. When you mention genomic embeddings, what sort of information is input into the model for representation? Some things that immediately come to my mind are somatic mutations, their genomic coordinates, rep and alt alleles, and etc., and gene expression level. Thank you.
So that's a great question. So for that particular approach, we were leveraging an institutional resource. So at Dana-Farber, we've had this thing called Profile going on for the last decade, which has been targeted next generation sequencing of somatic sequencing only. And so the inputs, therefore, are somatic mutations, somatic copy number alterations, and the associated metadata that goes with it. We would love to have it be more expanded, right? So either whole exome sequencing or exome and RNA sequencing. We've recently moved towards clinical exomes and RNA-seq. But for this study, because we have almost a decade's worth of panel sequencing with the clinical data, we've had to restrict ourselves to that. But even what's been curious to see is that even in those settings, these latent spaces end up recovering clusters that are surprising to us, that yield things that are not obvious, right? And so, the more subtle relationships, for instance, of like cyclin D1 amplifications in ESR1 mutant treated breast cancer are things that are emerging concepts, but one that we can recover independently, that are clusters that are in spite of not really knowing anything about it.
Okay, another question is online. When incorporating mutational features into your biologically guided AI models, have you explored using continuous variant allele frequencies VAP values instead of binary mutation states 0 or 1? If so, did it improve model interpretability or predictive performance? That's the question.
Yes, that's a great question. And so I think what you're getting at is can we, with the data we have, effectively try to do a better job of assigning clonality to the mutations that we see, so clonal or subclonal, and use VAF or cancer cell fraction or something like that as the better measurement. Unfortunately, these targeted tumor-only somatic panels are not the cleanest data sets to do this in. And so we've done some pilot experiments trying to do this now with some of the other data sets where we can get better, higher confidence allele frequencies or cancer cell fractions to do that kind of work. To be fair, it hasn't actually made a big dent in some of the inference we're doing. I don't think that's because there's no real biology there that would be meaningful. I suspect it's more of a power thing, right? We want, if we're looking, if that's getting towards, subtleties regarding clonality, you need and you're wondering about, the subclonal mutations and how they might contribute in a, localized or advanced population. You're starting to get down to such small numbers for any given use case. It's going to be hard to feel confident. So I think it's actually real. It's a great idea. I think we're limited in terms of our cohort size to make the most of it.
Okay, one more question. I assume. Okay, sorry. I assume you may have tested different combinations of patient level features for example, genomic versus transcripting versus clinical variables rather than using all available data at once. Which combination of modalities or features provided the most robust and biologically meaningful predictions in your experience?
Really great question. I think it depends on the question we're asking. So for some of the more focused questions, having expanded somatic data along with transcriptional data ended up being very useful. Again, because for the sort of the patient similarity generalized approach, the goal there being that I hesitate to use the word foundation model. I don't really think it really applies there, but it's sort of could be used for things that are not necessarily what it was intended for. We don't have data sets big enough to do anything other than that somatic panel data to do anything other than what I described. So it's hard to get a sense of features. That said, we do have some projects underway for to address certain kinds of dimensions there. So for example, I have a student who's actually just presented on this morning in our lab meeting, some worked up progress of you can take the tumor-only panel data, and this is through a collaborator at Dana-Farber named Sasha Gusev, and impute the germline information from those tumor-only panels. Then you can calculate polygenic risk scores from the imputed germline data and use that as a proxy for, representing the germline. And then you can combine germline information and somatic information and ask, for let's say certain kinds of outcomes across the board, which features matter more. Maybe not surprisingly, for a lot of the sort of downstream like therapeutics or overall survival things, the somatic information has proven to be much more useful than germline. But again, it's early days. And the germline maybe is far more relevant for oncogenesis, for instance, which would not be surprising either. But for some of these other modalities like DNA and RNA and whatnot, I think there's just remains just a shortage of volume of data to feel confident that we have big enough data to take that question head on. Even the big biobanks that are arising, they're largely one modality, right? And so or they're not big enough. So like HMF, which has, genomes and transcriptomes, has maybe 8,000 genomes, 8,000 patients total. And then you spread that across a bunch of different cancer types, a bunch of different contexts. It's just underpowered.
Okay. So I'd like to ask you one more question. I'd like to ask about the sample size. So to make better expectation, but better prediction. So, but I think that the size would be the bigger the better. But how do you think is the minimum size of the patient information for the making some model? So if the other court, other country will try the similar model. So how much the sample is needed for the make some?
Well, that's a great question. I think I think it depends on in the end depends on what you're trying to do, right? To our surprise, we only needed 1,000 prostate cancer exomes split between for one clinical variable to find something we thought was interesting in that one first example.
Okay.
In the second example, for these generalized sort of similarity methods, we currently have about 20 plus thousand patients, and we know that's woefully insufficient. It's not enough. And if you look at sort of where other domains and fields have gone, the most extreme examples, right, with like language models, if they've got trillions of words, and we are so far away from that right now in biology generally, much less cancer biology. That may not be that's why it's not surprising that those single cell foundation models, which sound impressive, oh wow, 100 million cells, that's a lot. It's not it's not enough. It's not even close to being enough. And then on top of that, the idea that we should represent the information in those complex models the same way that people are making architectures for language also probably isn't right. And so we need some creativity on the modeling side as well. And so the short answer is more is always better, but sometimes you don't need as much as you think you do. I've been pleasantly surprised sometimes, but mostly disappointed.
And your model, so besides the molecular information, the clinical information is also important. But I'm not sure in how much how different in Korea and United States. In Korea, it's common to take get some clinical information from the hospital because every hospital has their own database or closed system. So if we integrate different clinical information from different hospital, it's very difficult. So how is the situation in United States? How can you handle that problem?
It is the exact same problem. It's the exact same problem. And in fact, we can see such subtlety. The issues are that issues is everywhere, and it's also part of a general machine learning challenge, like about out of distribution or domain shift, or making sure models generalize across populations or contexts is a chronic problem for which there's a variety of attempts at solutions. But we think the only way to really solve this is that there needs to be creative ways to get hospitals and systems to pool their data together so that people can create generalized models. In the United States, that's hard because there's lots of protections around privacy, and if you're talking about getting unstructured, like entire electronic health records, the lawyers in the hospital are not going to let you just share it easily. So we have some early partnerships via these federated learning platforms, which are supposed to let us share things, let us deploy models in each hospital without having to share the data and keep privacy at stake. But even that is debatable. I think there are some machine learning people who think that there are ways to crack those platforms, and it's complicated.
Okay. One more question is listed, uploaded in the internet. So the question is this: in your model for metastatic prostate cancer, do you observe any context-specific molecular features that differ between primary and metastatic sites? And how does the model handle such spatial or temporal heterogeneity?
So that's a great question. And for that particular test, we didn't even have enough clean information about, for instance, metastatic site. So it wasn't even a consideration, and so we were blinded to that. And but we acknowledge that that is one of many kinds of confounders and concepts that would make for a far more effective model. That was basically a toy model in the simplest setup we could come up with just two kinds of molecular readouts and one very simple clinical classification task. So you're spot on that there's a lot of other ways to make these kinds of approaches better, smarter, faster.
Okay. One more question. The question is this: how effective do you think such omics interactive models could be for less genetically predisposed conditions like metabolic conditions such as diabetes, etc.?
So I think there, it depends on what the use case is, but there's a couple ways. I mean, so you could, for example, like what my student's trying to do is, calculate polygenic risk scores for people who have these common diseases that may not be directly cancer related, but perhaps have some bearing on what happens to you if you get cancer. That's turning to be messy and not clean. Another way to do this would be to make sure that you have basically the access to the entire clinical record, and you can really cleanly infer who has diabetes or who has heart disease or who has rheumatoid arthritis, and does that matter in certain contexts, and do that at scale. Again, those historically have been challenged, but at least within a given hospital, like we now can. One of my colleagues, Ken Keel, who has a nice set of research programs in Data-Farber around deploying language models to extract and learn those information cleanly, and so that we can get that information at scale across 50, 100, 500,000 patients quickly, so we can do analysis. And so, but again, even then the issue is how does that generalize to another hospital down the street, much less across the country or across the world. And so I think we're a little bit of a ways away.
Okay, thank you very much. I think there is no more questions, so I think we should close this session. So thank you very much for your wonderful talk.
Thank you.
Thank you.